So here I'm going to talk about chemical reactions. This is one of the last topics of this unit. Chemical reactions are where atoms exchange electrons between them. Almost any time electrons move from atom to atom, there's some kind of an energy exchange that takes place. And if the energy exchange is in the negative, the energy is released away from the atoms and away from the electron. If it's in the positive, the atoms and the electron absorb energy from the surroundings. Um, Here we got a thermite reaction taking place, which is the reaction between aluminum and usually an iron oxide. It's very violent. It's just rust and aluminum, but it burns so hot it'll burn underwater. And all that's happening here is electrons are exchanging between atoms. Here we have electrons exchanging between hydrogen and oxygen, which produces the thrust that pushed the shuttle Atlantis off the launch pad. Chemical reactions are what make your life possible. Without them, your body would just be an inanimate lump of chemicals. You basically eat foodstuffs to break down the chemicals to do two things with them. Either they become part of your body or you burn them for fuel. Now, we represent chemical reactions with chemical equations. So here's a really simple one. This says carbon monoxide plus oxygen yields carbon dioxide, and it shows an equilibrium arrow here, although generally speaking, it goes to the right. So carbon monoxide and oxygen react to form carbon dioxide. The heat exchange for the reaction is a negative 283 kilojoules per mole. What that means is, since it's negative, that when carbon monoxide and oxygen come together, the electrons exchange around and they form carbon dioxide, and energy is given off per carbon monoxide mole, and per mole of carbon dioxide produced, 283 kilojoules is given off, which is a lot. This is an important reaction because this is the reaction that makes automobile exhaust safe. This also makes fireplace gas safe so that you won't die of carbon monoxide poisoning. Now, the enzymes that are found in the human body are the workers that make the chemical reactions that happen in the human body happen at reasonable condition. In other words, you cannot afford to burn your energy, food energy, this way. Your body would incinerate. You need to burn it in a more reasonable way. Enzymes are the chemicals that make this happen. They're called catalysts. What they do is they make chemical reactions possible, and not only that, they speed them up. Most of your enzymes are actually proteins, and you're making them all the time. To accelerate chemical reactions, an enzyme actually lowers the activation energy of the reaction. This is a three-dimensional rendering of a human enzyme called glyoxylase, which basically detoxifies what are called aldehyde groups on biochemicals. You've got these enzymes in your liver where the liver, as it's processing foods that you've eaten from a meal, the aldehyde groups can be processed so that they don't have any toxicity. So, what does an enzyme actually do to lower activation energy? Well, what they do is they bind to substrates with what is called their active site. This is the active site of an enzyme. This gray ball here is an enzyme. That's the active site. The active site has a specific shape. So an enzyme can only bind to a certain substrate. It cannot bind to any substrate. And what that does is it forms a complex. That's this. This is the enzyme substrate complex when they're bonded together. And that allows then the enzyme to make the electron arrangement here a little different. And in this case, it makes these two chemicals fly apart and they go their separate ways. So this reaction diagram here kind of represents what we call the induced fit model of enzyme action, where an enzyme bonds to a substrate at the enzyme's active site that stresses the shape of the substrate. Okay, they call that the induced fit activity there, where they don't quite perfectly match until they bond together, and then the enzyme and the substrate together meld. At that point, then, the electronic arrangement inside the substrate is altered, and then the products are released. This is a graph that describes what's happening in this reaction. If X is the green structure and YX are these two chemicals that are being released, or I'm sorry, if X is the green structure, and Y is the blue and red product. What we're saying here is that to turn X into Y, you have to add a lot of energy. That's called the energy barrier, the energy hump, and then Y will form. Now, how does an enzyme, work in this? Well, what the enzyme does is it brings this chemical into a configuration so that it can more easily become the products, which are the blue and the red product here. It lowers the energy barrier. That's what the red dotted line is. The enzyme actually binding to the substrate at the active site changes the shape of the enzyme, changes the shape of the substrate a little bit. It changes the electron configuration slightly inside a couple of the atoms, and as a result, products are released that were different than the substrate coming in. Without the enzyme, it would take a tremendous amount of energy, that's this big hump, for the green thing to just spontaneously collide with something and form the blue and the red product. So this right here, this graph is showing an endothermic reaction, meaning that the reactants have less energy than the products. Products are higher energy than reactants. If it had been the opposite, okay, and X was up here and Y was down here, then it would be an exothermic reaction. Thermites like that. This is an exothermic reaction. Combustion of hydrogen gas and oxygen, exothermic reaction. This particular reaction, endothermic reaction. Endothermic reactions don't release heat and light, they absorb heat and light. It's the opposite. Now these animations kind of try to demonstrate what an enzyme does. This is an enzyme and that's its active site, bonds to the substrate. The little stray molecule provides the little oomph necessary for the substrate to crack in half. Water is used to mend the electron orbitals that are open when the product gets cracked in half. So that's why water is consumed. Water is made of H2O. So the H of a water molecule covers one broken bond and the OH covers another broken bond. Over here, we have two pieces locking together at the active site of the enzyme. Stray molecule hits them. Boom, the stray molecule provides the oomph to lock these two molecules together, but then a water molecule is released because an H and an OH was pulled off right in this little junction area there. Now, enzymes are not used up by chemical reactions. They only speed them up. So you'll notice the enzymes are not being used up. The substrate's being changed into products, but the enzyme's not being changed at all. Enzymes do denature if you heat them up too much. So if you boil some chemicals with an enzyme in it, the enzyme is rendered useless. It won't work anymore. And what's being demonstrated here is the induced fit model of enzyme action. It holds that enzymes bind to substrates and change their 3D shape. The induced fit between the enzyme and substrate weakens the bond within the substrate, and when those bonds are weakened at the active site, the products can be made then. So enzymes are very specific. They can only bind to specific substrates and form then specific products. So enzymes need hundreds, if not thousands of enzymes. Each cell in your body needs, if not hundreds, thousands of enzymes to perform all the chemical reactions they got to perform. It'd be great if one enzyme would do everything, but it can't. So here what we have is hydrolysis. Hydrolysis is the action of an enzyme chopping a polymer into monomers. It basically gets in between the monomers, stresses the bonds so that the monomers break apart. Water is used to fill the orbitals of the broken apart monomers. Over here we have condensation, where an enzyme is actually bringing monomers close to each other and then it bonds them together and it spits out water as a product. These two reactions are opposites to each other. You do this in your digestive tract when you eat food, and you do this with the monomers from your food coming from your digestive tract to build them back up into your own bodies. So this might be cow protein, and this is human protein being made. Cow protein being hydrolyzed, human protein being manufactured. Okay, and obviously you eat proteins from other animals and you make human proteins out of them. And these animations kind of describe that. On the lower left here, I'm actually showing you the atoms. These are amino acids. This is a protein here. And the enzyme's actually cutting the amino acids apart from one another to turn one polypeptide into 6 or so different amino acids. This, on the other hand, is similar to this reaction above it. These are monosaccharides. And what the enzyme is doing is it's pushing a few atoms off between them and making them lock together. So you'll see there's a bridge here between the atoms. Now, that kind of concludes the energy part and the enzyme part of our molecular biology unit. The last thing I just need to mention here is food labels. We're going to talk a little bit about these in class. You need to be able to read food labels. Food labels will tell you how much fat, how much protein, and how much carbohydrate are in a food, as well as how much salt. A typical Coke Classic has more than 40 grams of sugar in a 12-ounce drink of soda, and you shouldn't even get 40 grams of that sugar really in a day. A single Big Mac has 30 grams of fat in a single burger, and that's about half of the fat that you should take in for a day. And a single package of ramen noodles has 1.7 grams of salt, which is most of the 2.4 grams that you're supposed to max out at per day. Out of a 2,400 calorie diet per day, you should have about 3,000 calories. You should have about 370 grams of carbohydrate, 155 grams of protein, and 66 grams of fat. Generally speaking, you never want to get much less than 50 grams of protein per day, because if you do, your muscle mass will decline in your body. You cannot maintain your muscle mass. You need at least 155 grams of protein. You'll hydrolyze that in your digestive tract into amino acids, and you'll take 155 grams of amino acids, and you'll use that to build up muscle in your body that's breaking down. If you don't get at least 50, your muscle will actually weaken in your body. So we'll talk more about food labels in class, and we'll probably do a project with them. Anyway, that concludes our discussion of energy, enzymes, and food labels. I will see you in class.